Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. Today, our topic of discussion is gene mutations, and in today's topic, we will be looking at the nature of mutation. And under this heading, we will look at the importance of mutation, categories of mutations, types of gene mutations, and mutation rates. So, first of all, the nature of mutation. We know that DNA is a highly stable molecule that replicates with amazing accuracy, but despite this amazing accuracy, changes in the DNA structure and errors of replication are inevitable and they do occur. And we can define a mutation as an inherited change in genetic information. The descendants may be cells produced by cell division or individual organisms produced by reproduction. So now coming towards the importance of mutations. So mutations are both sustainers of life and the cause of great suffering. So mutation can affect in two ways. It can cause changes that are sustainable for life, and they can also cause great suffering. So on the one hand, mutation is the source of all genetic variation, and genetic variation is the raw material of evolution. So without mutations and the variation that they generate, organisms could not adapt to changing environment, and if they will not adapt, they would risk extinction, mean that they can disappear from the world. So on the other hand, most mutations have detrimental effects, and mutation is the source of many human diseases and disorders. And much of the genetics focuses on how variants produced by mutations are inherited. So genetic crosses are meaningless if all individuals are identical homozygous. They are identically homozygous for the same allele. So if they are identically homozygous for the same allele, then there is no use of doing crosses, because if they are homozygous, their progeny will also be homozygous. So mutations serve as important tools of genetic analysis. The solution to almost any genetic problem begins with a good set of mutants. So if good set of mutants is not available, then it is difficult to solve a genetic problem. So much of the Mendel's success in unraveling the principles of heredity can be traced to his use of carefully selected variants of the garden pea plants. So similarly, T.H. Morgan and his students discovered many basic principles of heredity by analyzing the mutant fruit flies. So if they would not have come to different variations of the organisms under study, they would have not discovered different rules of heredity, which are nowadays basis of the whole genetics. So for example, if you look at the phenotype of flies, this is a wild type fly, and when mutations arise in different genes, they give rise to different phenotypes. For example, bar eyes, flies with bar eyes, flies with miniature wings, flies with white eyes, and flies having curved wings. Similarly, kidney phenotypes, vestigial wings, curly wings, and many other phenotypes. So variation is the source of basis for the genetic studies. So now coming towards the categories of mutation. So we know that in multicellular organisms, we can distinguish between two broad categories of mutations, and they are somatic mutations and germline mutations. So somatic mutations arise in somatic cells, and somatic cells are those cells which do not produce gametes, and these mutations are passed on to other cells through the process of mitosis, which leads to a population of genetically identical cells, and that genetically identical population of that genetically identical cells is also known as a clone. The earlier in development that a somatic mutation occurs, the larger the clone of cells within that individual organism that will contain the mutation. So if we look at this figure, uh, we can see that in the case of somatic mutations, when a somatic mutation is occurring, then this somatic mutation will result finally in a patch of color on the skin of fish after meiosis. But in the case of germline mutations, if a mutation that was a somatic mutation in first case, if this mutation is a germline mutation, then it will make the whole fish red. So this is the main difference. So when a mutation is occurring in the somatic cells, it will only make a patch of cells, a clone of cells, a population of genetically identical cells. But if that mutation is a germline mutation, then this mutation will affect the whole skin of the fish. So because of the huge number of cells present in a typical eukaryotic organism, somatic mutations must be numerous. For example, there are about 10 raised to power 14 cells in the human body. If a mutation arises only once in every million cell divisions, which is a fairly typical rate of mutation, hundreds of millions of somatic mutations must arise in each person. So the effect of these mutations depends on many factors, and these factors include the type of cells in which they occur and the developmental stage at which they are arising. So many somatic mutations have no obvious effect on the phenotype of the organism because the function of the mutant cell is replaced by that of the normal cells, and sometimes even the same cell uses another method to overcome that mutation. So however, cells with a somatic mutation that stimulates cell division can increase in number and spread, and if mutation occurs in these genes which are used in cell division, so then this type of mutation can give rise to different cells with a that have a selective advantage, and they will start growing uncontrollably, and this is the basis of all cancers. So when mutations are present in the genes that are coding cell division, that are coding different proteins which are involved in cell division, then it will be disastrous finally for the organism carrying that somatic mutations. So now coming towards the germline mutations, and germline mutations arise in cells that ultimately produce gametes, and these mutations can be passed to future generations, producing individual organisms that carry mutations in all their somatic as well as germline cells. When we speak of mutations in multicellular organisms, we are usually talking about the germline mutation. So in single cell organisms, however, there is no distinction between these germline and somatic cells, because when cell division takes place in unicellular organisms, then division results in new individuals. So one cell divides and two new individuals are produced. So historically, mutations have been partitioned into those that affect a single gene, and they are known as gene mutations, and those mutations that affect the number or structure of chromosomes, they are known as the chromosome mutation. So this is the difference which is being used historically to indicate the mutation. So this distinction arose because chromosome mutations could not be observed directly by looking these chromosome mutations can be observed directly using microscopy, whereas gene mutations could not be detected by microscopy, and they can only be detected by observing their phenotypic effects. So now with the development of DNA sequencing technologies, gene mutations and chromosome mutations are distinguished somewhat arbitrarily on the basis of their size of the DNA lesion. So nevertheless, it is useful to use the term chromosome mutations for large scale genetic alterations that affect the chromosome structure or the number of chromosomes, and the term gene mutation is used for a relatively small DNA lesion and that affects a single gene. So this is a difference between the chromosome mutations and gene mutations. So now let us move toward the types of gene mutations. So there are a number of ways which are used to classify the gene mutations. So some classification schemes are based on the nature of the phenotypic effect, whether the mutation alters the amino acid sequence or the protein, and if so, how it occurs. So other schemes are based on the causative agent of the mutation, and still uh, other methods focus on the molecular nature of the defect. So most appropriate scheme depends on the reason for studying the mutation. So we should have a reason, and then we can classify the mutations according to our reason for study. So but here in our course we will categorize mutations primarily on the basis of their molecular nature, but we will also encounter some terms that relate the causes and phenotypic effects of mutations. So first type is base substitutions. So base substitutions, the simplest type of gene mutation is a base substitution, and base substitution is actually the alteration of a single nucleotide in the DNA. So because of the complementary nature of the two DNA strands, we know that DNA is double stranded and both strands are complementary to each other, and when the base of the one nucleotide is altered, the base of the corresponding nucleotide on the opposite strand will also be altered in the next round of the replication. So a base substitution therefore usually leads to a base pair substitution. So and base pair substitution as you can see in this figure, that if this is a this first panel, left panel is original DNA sequence, and in the right panel a base substitution is shown. It means that, for example, this is third codon is GTA, and in this sequence T has been changed to C. So this is a base substitution of in single codon, and this only alters a single codon. And base substitutions are also of two types. One type is transition, and in transition a purine is replaced by a different purine, or alternatively a pyrimidine is replaced by a different pyrimidine. And second type is transversion, and in transversion a purine is replaced by a pyrimidine, or a pyrimidine is replaced by a purine. So the number of possible transversions is twice the number of possible transitions, but transitions are frequent than the transversions. So in this figure we can see that in the case of transitions, purine molecule is replaced by a purine, and similarly pyrimidine is replaced by another pyrimidine. But in the case of transversions, purine is changed into pyrimidine, or pyrimidine is changed into purine. So there are only 4 different combinations, but in the case of transversion there are double options for transversions, but transitions are more common. So another type is the insertions and deletions. The second major class of gene mutations contains insertions and deletions, the addition or the removal of one or more nucleotide pairs. So although base substitutions are often assumed to be the most common type of mutations, molecular analysis has revealed that insertions and deletions are more frequent. And insertions and deletions within sequences that encode proteins may lead to the frameshift mutations and changes in the and this frameshift these frameshift mutations, they change the whole reading frame of a gene. So insertion deletions are mostly causing the frameshift mutations, and when they are present in the coding regions, they will change the whole reading frame of the protein, and whole sequence of the protein will be disturbed. So when we look at this figure, so if a nucleotide is inserted, for example, this is the original codon GTA. So a T is inserted. After that, first T, then this will become GTT, and A will move to the next codon. And similarly, if this T is deleted from this codon, then the other two bases, the other base, next base will be part of the, for example, this G will become the part of codon, and in both ways reading frame will be changed and structure of the protein will be disturbed. So the initiation codon in mRNA sets the reading frame, and this thing is mostly studied in the molecular biology. After the initiation codon, other codons are read as successive non-overlapping groups of 3 nucleotides. The addition or deletion of a nucleotide usually changes the reading frame, altering all the amino acids encoded by codons following the mutation. So many amino acids can be affected. So frameshift mutations generally have drastic effects on the phenotype. So not all insertions and deletions lead to frame shifts. However, because codons consist of 3 nucleotides, insertions and deletions consisting of any multiple of 3 nucleotides will leave the reading frame intact, although the addition or removal of one or more amino acids may still affect the phenotype, but there will be no frame shift. And those mutations that are multiples of 3 nucleotides, whether they are insertions or deletions, they will not change the reading frame, although they can result in drastic effects. So these mutations are called in-frame insertions or deletions. Now coming towards the expanding trinucleotide repeats. So in 1991, an entire novel type of mutation was discovered, and this mutation occurs in a gene known as the FMR1, and it causes Fragile X syndrome. And the most common hereditary, and this is the most common hereditary cause of mental retardation, which is nowadays known as the intellectual disability. And the disorder is named because of this change in the X chromosome. So in specially because in specially treated cells of persons having the condition, the tip of the X chromosome is attached only by a slender, a weak thread. So FMR1 gene contains a number of adjacent copies of trinucleotides, and they are CGG. The normal FMR allele, which is not containing the mutation, has almost 60 or fewer copies of this trinucleotide, but in persons with Fragile X syndrome, the allele may harbor hundreds or even thousands of copies of the nucleotide, 3 nucleotide repeats. So mutations in which copies of a trinucleotide may increase greatly in number are known as the expanding trinucleotide repeats. And as you can see in this figure that this is a affected chromosome, X chromosome. This is a normal X chromosome, and the chromosome containing mutation has a small thread-like things on long arm of the chromosome, and this is caused because when multiple repeats are present, then because of these multiple repeats, DNA methylase enzyme is attracted, and this methylase enzyme methylates the cytosine residues in the DNA, and because of this, this region is containing the repeats is condensed, so this becomes inactive. So if the product of this gene is this normal gene is run on the gel, it is giving the product, but if in the case of abnormal repeats, this product is not visible on the gel. And there are several disorders which are caused because of the increase in the number of repeats. For example, spinal and bulbar muscular atrophy and Fragile X syndrome, Jacobson syndrome, and many other syndromes. And we have also studied Huntington's disease previously. This is also because of the expansion of 3 nucleotide repeats. So how an increase in the number of nucleotides produces disease symptom is not yet known, and in several of the diseases, for example Huntington's disease, the trinucleotide CAG expands within the coding part of a gene, and this produces a toxic protein that has extra glutamine residues. And in other diseases, for example in the case of Fragile X syndrome and myotonic dystrophy, the repeat is outside of the coding region of the gene, and therefore they must have some other mode of action. So at least one disease which is rare type of epilepsy has now also been associated with an expanding repeat of 12 base pair sequence. And although this repeat is not a trinucleotide, but it is a multiple of this repeat is a multiple of 3 nucleotides. Therefore this disease has been included in this list of nucleotide repeats. Now the mechanism that leads to the expansion of trinucleotide repeats is not still not clear, and strand slippage in the DNA during replication and crossing over between misaligned repeats are two possible sources of the expansions. And single stranded regions of some trinucleotide repeats are known to fall into hairpins during the replication, and other special DNA structures when they are in the form of hairpins, the DNA polymerase becomes confused, and then different repeats are added. So such structures may promote strand slippage in replication and may prevent these errors from being recognized, because there are some different procedures and mechanisms which are used to repair or correct the problems or mistakes in the DNA sequence. But when these types of hairpins are other structures are formed, special structures are formed, then the mechanisms used to repair those problems are not working, and in this way they can result in the replication errors. So this figure is also telling us the slippage, strand slippage in replication. So in the case of when replication starts, this is the normal, sorry, at the start of replication, these are the 8 repeats, and then when a hairpin loop is formed, then uh, this is causing the increase in the length of the repeats. So now this has 13 repeats, and because of this slippage, this number has increased from 8 to 13. So next is the phenotypic effects of mutations. Mutations have a variety of phenotypic effects, and the effect of a mutation must be considered with reference to a phenotype against which the mutant can be compared. So if we don't have a reference phenotype, then we cannot compare. So with which is usually the wild type. The reference phenotype is mostly the wild type phenotype, and this wild type phenotype is the most common phenotype that is naturally present in the population of organisms. For example, if we look at the example of Drosophila, most Drosophila melanogaster in nature have red eyes. So the red eyes are considered as the wild type eye color, and any other genetically determined eye color in fruit fly is will be considered as the mutant phenotype. So a mutation that alters the wild type phenotype is known as the forward mutation, whereas a reverse mutation changes a mutant phenotype back to the wild type phenotype. So this is the difference between forward and reverse mutations. And there are special terms that are used to describe the phenotypic effects of mutations. And first one is a missense mutation. And missense mutation is a base substitution that alters a codon in the mRNA, resulting in a different amino acid in the protein. And second is a nonsense mutation, and it changes a sense codon which is specifying an amino acid into a nonsense codon that is specifying a termination codon during the translation process. Uh, if a nonsense mutation occurs early in the mRNA sequence, so the protein will be greatly shortened and will usually be a non-functional protein. And next type is a silent mutation, and silent mutation alters a codon, but thanks to the redundancy of the genetic code, the codon still specifies the same amino acid, because redundancy of the genetic code means that there are multiple codons for a single amino acid. So there are 20 amino acid, but there are total 61 codons that are specify different amino acids, and there are 3 stop codons. So a neutral mutation, next is a neutral mutation. A neutral mutation is a missense mutation that alters the amino acid sequence of the protein but does not change its function. So neutral mutations occur when one amino acid is replaced by another that is chemically similar, or when the affected amino acid has little influence on the protein function. Then it is known as the neutral mutation. So in this figure, we can see 3 different types of base substitutions. First one is the missense, then nonsense, and then silent mutations. In the case of missense mutations, um, if this is a normal sequence of DNA, mRNA, and proteins, in the case of missense mutation, if C in this sequence is changed to T, then this will result in leucine in the protein sequence. And if this this C is changed into A in the case of nonsense, then this will result in creation of stop codon in the mRNA, and this will result in termination at the time of translation. But if the normal A nucleotide, adenine nucleotide in the DNA sequence is changed to G, then this will be a silent mutation because this codon is also coding the serine amino acid, which is the normal amino acid. So these are different mutations. Now coming towards the loss of function mutation. Loss of function mutation cause the complete or partial absence of normal function, and a loss of function mutation therefore alters the structure of a protein, that the protein no longer works correctly, or the mutation can occur in regulatory regions that affect the transcription, translation, or splicing of the protein. And loss of function mutations are frequently recessive, and diploid individuals must be homozygous for the mutation before they can exhibit the effects of the loss of functional mutation, loss of functional protein. And another type is a gain of function mutation, and a gain of function mutation produces an entirely new trait, or it causes a trait to appear in inappropriate tissues or at inappropriate times during the development, and these mutations are frequently dominant in their expressions. So single allele is enough for causing the disease. Still, other types of mutations are conditional mutants, and in conditional mutants which are expressed, these conditional mutations are expressed only under certain conditions, and lethal mutations, they cause the premature death of the individual. So in the case of lethal mutations, um, the individuals do not they are not when the lethal mutations are present, individuals will die prematurely and they will not be born normally. So next is suppressor mutations. A suppressor mutation is a genetic change that hides or suppresses the effect of another mutation. So this type of mutation is distinct from the reverse mutation, in which the mutated site changes back into the original site, original wild type sequence. A suppressor mutation occurs at a site that is distinct from the site of original mutation. So thus an individual organism with a suppressor mutation is a double mutant because it possesses both mutations, the original mutation and the suppressor mutation, but exhibits the phenotype of an unmutated wild type. It has two mutations but its phenotype is normal. So this is in the case of suppressor mutations. So for example, the normal eye color in Drosophila is red color. The red color is normal wild type eye color, but a mutation in the A allele makes it white. So if this A minus is converted back to A plus, then this will be a reverse mutation. But if a second mutation B minus is causing the color to be red, then this will be a suppressor mutation because A allele is minus, and now with this A minus, a B minus allele is produced, and this is acting as a suppressor of the mutation. So this is the basic difference. When an allele is changed back into the normal allele, then it is reverse mutation. But when another mutation in the same gene is causing the wild type mutation, then it is the suppressor mutation. So an intragenic suppressor mutation occurs in the same gene that contains the mutation being suppressed. So in this figure we can also see that if in the protein normal sequence is leucine, but when a mutation occurs it produces phenylalanine, and when another mutation at second site occurs, it again produces the normal protein. So this is just like Drosophila, the example of Drosophila, this is producing the normal protein. So this will result this is an intragenic suppressor mutation which is producing the wild type phenotype. So we can also see this. For example, this is a normal sequence of DNA and encoded protein. So one nucleotide deletion will result in creation of a stop codon, premature stop codon. But when a duplication occurs, then this duplication restores the uh, length of the protein. Although this is this amino acid is different from the um normal amino acid, but it is restoring the sequence of the protein. So this is also an intragenic suppressor mutation. So the second form is of suppressor mutation is intergenic suppressor mutation, and intergenic suppressor mutation is actually when a mutation in another gene is restoring the mutation of another gene, then this will be intergenic suppressor. So in this example, we can see that in the first panel in the A figure, a normal protein, fully functional protein is produced. But when a mutation occurs, then this mutation is producing a premature stop codon, and a shortened and non-functional protein is produced. And when a mutation occurs at the second site in the second gene, then this mutation is occurring in a tRNA molecule, the tRNA molecule that was previously carrying a different amino acid. But when this mutation has occurred, this has changed the anticodon, and that anticodon is now pairing with uh, the stop codon, which was produced in the protein sequence because of another mutation. So now this tRNA is pairing with the stop codon, and a fully functional protein is produced. So this is the second type of suppressor mutation, and is intergenic suppressor mutation. So in this table, the characteristics of different types of mutations are given. So we have already studied most of these things, and you should keep in mind all these definitions of different types of mutations. So now coming towards the mutation rates. Uh, the frequency with which a gene changes from wild type to a mutant is referred to as the mutation rate, and is generally expressed as the number of mutations per biological unit, which may be mutations per cell division, mutations per gamete, or mutations per round of replication. So for example, the mutation rate of achondroplasia, which is a type of hereditary dwarfism, is its mutation rate is about 4 uh, mutations per 100,000 gametes, and usually it is expressed more simply as 4 into 10 raised per minus 5. So in contrast, mutation frequency is defined as the inheritance of a specific type of mutation within a group of individual organisms. So for the achondroplasia, the mutation frequency in the US population is about 2 into 10 raised per minus 4, which means that about 1 in 20,000 persons in the US population is carrying this mutation. So there is a difference between mutation rate and the mutation frequency. This should be clear. And in this table, different rates of mutations are given for different conditions in different species. For example, if in the case of, if we look at the humans, in the case of Huntington's disease, per gamete rate is 1 into 10 raised per minus 6, and similarly other examples of other disorders are also given. So this is the first part of mutations, and hopefully in the next chapter we will finish this topic. So thank you, and see you in the during the online session.